Hello dear students, welcome to Genesis. Well, let us start the video by understanding what is fertilization. Well, fertilization is the fusion of the male gamete, which is sperm, and the female gamete, which is ovum. So at the time of copulation, a male releases close to 400 million sperms in the female genital tract. So this is basically released somewhere near the cervix, and now the sperm, being the motile gamete, will have to move from the cervix to the fallopian tube. So the rate with which a sperm moves is about 1.5 millimeter per minute, and then it reaches the fallopian tube where the egg waits for fertilization. Now the egg is released on the 14th or the 13th day of the menstrual cycle. So after the 13th or the 14th day, if there is copulation, then the chances of pregnancy increases. So now let us understand the structure of the male and the female gamete. Well, the male gamete is divided into three parts. It has a head that contains a nucleus and a bag called acrosome. Now this bag contains the enzyme called hyaluronidase. Now this enzyme is required for the sperm to melt the various layers of the egg. When I explain the structure of the egg, it's going to be all the more easier for you to understand. The second half is called the middle piece, and this middle piece is loaded with mitochondria, and this is that area which produces energy required by the sperm to move. The third and the very important part of the structure is called the tail, and the upper thicker part of the tail is known as the axial filament. This part, or axial filament, and the tail helps in the propulsion of the sperm. Let's look at the structure of the female gamete, which is ovum or egg. Now this ovum has three protective layers, not one but three. The outermost layer is made up of cells, and this layer is known as corona radiata. So these are the cells of corona radiata. The second layer is called the zona pellucida. So this basically is the zona pellucida of the egg, and the third and the innermost layer is known as the vitelline membrane, and inside the vitelline membrane is the nucleus. So here is the nucleus of the sperm, and this is the nucleus of the egg. Remember one thing, when we talk about fertilization, fertilization means fusion of the male and female gamete, and this fusion will be considered complete only when the nucleus of the sperm fuses with the nucleus of the ovum. Otherwise it will not be called fertilization. So the sperm will actually have to come and melt the three layers of the ovum and reach the nucleus so that it can fuse its nucleus with the nucleus of the ovum. Now as you already know, the sperms are of two types, okay, depending upon the chromosome that they carry. So the nucleus of the sperm and the nucleus of the ovum, they are haploid. That means they have half the set of chromosomes, and in case of sperm, they contain 22 autosomes plus one X chromosome. So this is one variety of sperm. Another variety of sperm will contain 22 autosomes plus one Y chromosome. But in case of female, the setup will always be 22 autosomes plus one X chromosome. So now imagine a condition: if this sperm goes and fuses the egg, then the child born will have one X and one Y chromosome and 44 autosomes. So the gender of the child is going to be male. But if this sperm goes and fuses with the egg, then the child will have 44 autosomes and 2 X chromosomes. So the gender is going to be female. Now at the time of copulation, when these sperms are released in the female body, remember one thing, these sperms can remain in the female body for a good span of time, for some couple of days, but their power to fertilize remains active only for 2 to 3 days. So if within 2 to 3 days they can reach an egg and fertilize it, then only it will result into the formation of the zygote, otherwise not. During fertilization, there can be many sperm who can manage to reach the egg. So like I have drawn one sperm here, actually there can be many sperms which can reach the egg, but the actual credit will go to that sperm which can melt all the three layers and will reach the nucleus. And remember one thing, if any one sperm can actually melt the layers and reach the nucleus, then the egg itself is going to produce certain chemicals which will not allow any other sperm to get inside further. This is done to prevent any scope of more than one sperm coming and fusing with the nucleus of the egg. Well, now let us understand what is implantation. Implantation is the process of fixing the blastocyst to the endometrium of the uterus. You already know the innermost lining of the uterus is called endometrium, but what we do not know is what is a blastocyst. Now, at the time of fertilization, when the sperm fuses with the ovum, what is produced is called a zygote. Zygote is unicellular, that means it is made up of a single cell, but this cell then undergoes certain mitotic division where they divide to increase in number. So this zygote will first divide, will undergo one single cleavage and will form 2 cells, then from 4 cells, 8 cells, 16 cells and onwards. Remember one thing, this fertilization takes place in the fallopian tube. So the zygote actually forms here, which is known as the ampulla of the fallopian tube. From here, the zygote will slip to the endometrium of the uterus and will get fixed there, and that process is called implantation. But during this journey where it is shifting from the fallopian tube to the uterus, it undergoes these mitotic division, and that final stage which goes and fixes to the endometrium of the uterus is not called zygote, it is known as blastocyst. What is the difference between zygote and blastocyst? Zygote is unicellular, blastocyst is multicellular, and it looks somewhere like this. So it has an inner lining of cells, it has a cavity inside. This structure is called blastocyst. So every time when you are defining implantation, you need to remember this stage, okay, that gets implanted to the endometrium of the uterus. It is blastocyst. Okay, now let us understand the difference between these three terms, zygote, embryo and fetus. Well, zygote is the structure which is unicellular and is formed right after fertilization. Next to this, when the zygote starts to undergo mitotic division, it forms a mass of cell which is known as embryo. Now this embryo extends from a gestation period of 3 weeks to 5 weeks. Gestation period is that entire period during which a baby develops inside the mother's body, which we know is of 9 months approximately. Now from the 3rd to the 5th week, this entire period is known as the embryo. Now first 3 weeks, the embryo is as small as the size of a pea and it does not develop anything that resembles human. So that is known as an early embryo stage. But by the time of 5th week of pregnancy, this embryo starts to develop heart and blood vessels. So that is known as the advanced stage of embryo. So this embryo is multicellular and this is how it is different from the zygote. And then comes the stage which is called fetus. Remember, fetus is that stage when the embryo now develops limbs and internal organs and it starts to resemble the human form. So this is where the internal organ and the major parts are formed. So understand the flow chart that will depict the various stages of development of human embryo. So the human embryo first starts with egg which gets fertilized with a sperm, and after that what is produced is called zygote. We already know it is unicellular. This zygote will multiply, divide and redivide to form a mass of cells which is known as the morula. This morula will further form a structure which is called blastocyst. How is a morula different from a blastocyst? Morula is a solid mass of cell, whereas when we talk about blastocyst, it has a cavity inside and the layer of the cell that surrounds that cavity is known as the tropoblast. And remember, this is that stage that implants in the endometrium of the uterus. Then is the next stage which is known as the embryo. The early embryo is only 3 weeks old and we know that it has no organs, no blood vessels in it. Next stage is the advanced embryo stage where it develops heart and blood vessels. Next is the fetus where it will develop internal organs and limbs and will start to resemble a human baby. And last stage, which is after 40 weeks, is the infant where the baby is given out of the mother's body. These are the various stages of development of human embryo. Now we come to that part where we are going to discuss about various extra embryonic membrane. Well, remember when the blastocyst goes and implants to the wall of the uterus, it develops few protective layers around it. These protective layers are called extra embryonic membrane. Their main function is to protect and to help nourish the growing embryo. So what are these? They are amnion. This is the innermost layer. That means the layer which is closest to the embryo. So if this is the embryo, the layer immediately after it, which surrounds it, is called amnion. And remember, the space between the embryo and the amnion is filled by a fluid which is called amniotic fluid. It's a very, very important fluid. The second to this layer is the chorion. So this is the layer which is called chorion, again protective in function. Third is allantois. Here is the third layer which is called allantois. Just like chorion, it is also protective in nature. Fourth is yolk sac. This is yolk sac. It does not have much function in human beings, but of course in birds and in other organisms it also plays some very important role like protection and nourishment. And the fifth one is the placenta, about which we are anyways going to study in details. Of all these, we have to study amniotic fluid and placenta in detail. So the amniotic fluid which surrounds the embryo serves certain very important functions for the embryo. What are those? Number one, it protects the embryo from any physical shock because it's a fluid, so it kind of dissolves the physical shock for the growing embryo. Second is it equalizes the pressure all around the embryo. It provides a restricted movement to the baby. And fourth, it prevents the sticking of the fetus to the amnion, which is that layer around it. So with this we finish today's lecture. In my next lecture I'm mainly going to cover placenta and twins. So meanwhile keep revising. Do not forget to subscribe my channel. Thank you.